What if, like, the sheer physical stiffness of your own body could literally shatter your DNA?
I mean, it sounds crazy.
It really does. Because, we usually think of cancer-causing mutations as this matter of, well, chemistry or physics. Like, we picture toxic chemicals from cigarette smoke binding to our cells, or ionizing radiation just blasting our DNA strands apart.
Exactly. We always think of mutagens as these external threats that invade the body.
Right. But today, for this deep dive, we are looking at a complete paradigm shift. We're exploring the idea that mechanical tension, just the physical rigidity of the microscopic environment around a cell, can somehow reach into the nucleus and rewrite your genetic code.
Yeah, it sounds almost like science fiction, honestly. The idea that just a physical squeeze could cause a genetic mutation. But we are looking at a really fascinating preview article published in the journal Cancer Cell today. It's authored by Nicole Cruz-Reyes and Derek Radisky, and they're basically unpacking this truly groundbreaking study by Hayward and a huge team of researchers. They've actually managed to map out the exact step-by-step molecular relay that converts physical force into genetic damage.
Which is it's a huge deal. Our mission today is to literally walk through this relay step by step. We're not skipping any mechanisms. We want to really understand how a stiff tissue becomes a mutated tissue, because this actually solves a massive medical mystery, right?
Oh, absolutely. I mean, for a long time, doctors have seen that fibrotic tissues, so that means scarred, dense, or, stiff tissues and chronically inflamed tissues, they carry a much higher risk of developing cancer.
Yeah, and that's true across multiple organs, right?
Right. But the bridge between those two concepts was just completely missing. Like, we knew inflammation created oxidative stress, and we knew fibrosis made the tissue scaffolding stiff. But how does a stiff scaffolding actually, create a mutation?
So to understand that missing link, let's shrink down and look at the cellular scaffolding itself. You call this the extracellular matrix, right?
Yeah, the ECM. So imagine it as this really complex three-dimensional web of proteins that basically holds your organs together.
Okay, I'm picturing a web.
Right. And in a fibrotic state, this web becomes heavily cross-linked. It gets super rigid. It completely loses its flexibility. Now, embedded right inside this rigid web are your epithelial cells, the cells that line the surfaces of your organs and ducts.
And, I mean, those cells don't have eyes. They can't see their environment. They only know what they can physically touch, right?
Exactly. They experience the entire world mechanically. So when the matrix around them stiffens, they literally feel it. And they feel it through something called ROCK-dependent actomyosin contractility.
Okay, let's unpack that because it's a mouthful. ROCK-dependent actomyosin contractility. So if you're listening and visualizing this, think of the cell having its own internal muscle fibers.
That's a great analogy, yeah.
Right. So the cell naturally tries to grip and pull against the scaffolding around it just to, maintain its shape and position. But because the scaffolding is suddenly totally unyielding and stiff, those internal cellular muscles, they have to pull harder and harder.
Right. The cell is just violently tensing up because it's trapped in this rigid cage.
That sounds exhausting for the cell.
It is. And that mounting physical tension inside the cell is compounded by a chemical signal. So while the cell is physically straining, it's also being bathed in inflammatory signaling, specifically an IL-6-JAK1 pathway.
Okay, IL-6.
Yeah, IL-6 is an inflammatory messenger driving a signal inside the cell via the JAK1 enzyme. So you have intense physical pulling combined with this inflammatory chemical environment. And together, these two forces basically hit a master panic button.
A panic button inside the cell. I love that visual. What does that actually look like biologically?
Well, it takes the form of a transcription factor, which is a protein that controls how genes are expressed. And this one is called STAT3. So the mechanical strain and the inflammation chemically activate STAT3.
Okay, STAT3 is the panic button. Got it.
Right. It travels right into the nucleus of the epithelial cell and forces the cell to completely change its behavior. The cell basically begins to pump out a cocktail of specific distress signals.
Like chemical SOS flares.
Exactly. The researchers identified them as TAM-recruiting chemokines, specifically things like CCL3, CCL5, CCL12, and CSF1. They are just broadcasting this distress out into the surrounding tissue.
Okay, wait, I want to pause here and make sure I'm getting the timeline right for this relay, because we just hit a lot of science.
Sure, let's recap.
So the epithelial cell is tensing up, it hits the STAT3 panic button, and it's blasting out these chemokine SOS flares. But at this exact moment, its DNA is still perfectly fine, right? The mutation hasn't actually happened yet.
Yes. That is a crucial distinction. At this stage, the cell is entirely healthy from a genetic standpoint. It is merely a stressed-out cell crying for help. The researchers proved this beautifully with genetically engineered models. When they selectively disabled that STAT3 panic button in the mammary cells, the entire deadly cascade just stopped dead in its tracks.
Because it couldn't call for help.
Right? No distress signals were sent, and critically, the DNA never got damaged, even though the environment was still stiff.
That is just wild. So to give a quick recap and summary for everyone listening before we move to step 2, bullet 1, a stiff tissue triggers physical cell tension and IL-6 inflammation. Bullet 2, this activates the STAT3 panic button. Bullet 3, the cell releases chemokines, those SOS flares, specifically CCL3, 5, 12, and CSF1.
Perfectly summarized.
It really makes you look at your own body differently, doesn't it? I mean, think about it. Have you ever considered how your individual cells might physically feel their surroundings before they decide how to chemically react? It's mind-blowing.
It's a whole new way of looking at cell biology.
Yeah. So, okay, the alarm's ringing, the distress signals, the chemokines are floating out there. Who actually answers the call?
Well, the signals are specifically tuned to recruit the immune system's first responders. They draw in a specialized type of immune cell called a CD163 positive tumor-associated macrophage, or TAM for short.
Okay, CD163 positive macrophages. Now, normally, macrophages are the good guys, right?
Absolutely. They're the protectors. They're basically the cleanup crew that arrives at a wound site to clear out debris, fight off infection, and just help the tissue heal.
Right. But this isn't a normal wound. So the macrophages arrive to help, but, and correct me if I'm wrong, they are stepping into the exact same rigid, stiff environment that caused the epithelial cells to panic in the first place.
Exactly. And this environment completely corrupts them. This is where we encounter a concept in the paper known as the mechanordox principle.
Mechanordox.
Okay.
Mechanics and oxidation.
Yeah. It's this fascinating mechanism where the physical compliance, like the actual stiffness of what a cell is touching, dictates its chemical output.
So how does the macrophage translate a physical touch into a chemical reaction?
Through its grip. When the macrophage arrives and tries to latch onto this highly cross-linked, super stiff collagen matrix, its grip sensors go into absolute overdrive.
Grip sensors.
Yeah. It uses adhesion molecules to hold on, and the physical resistance of the stiff wall triggers a very specific pathway. It's FAK-dependent, that's focal adhesion kinase, which reacts to the physical grip.
FAK.
And that FAK activation relies on an enzyme complex called NOx-NADPH oxidase.
Oh, I've heard of NOx.
Yeah, and this complex is entirely dedicated to generating reactive oxygen species, or ROS.
Wow, okay, so it's literally like calling the fire department to a burning building, but the extreme heat of the neighborhood instantly brainwashes the firefighters.
That is a terrifying but very accurate analogy.
Right. Just by touching the stiff walls, these macrophages are reprogrammed, they drop their hoses, pick up flamethrowers, and just start churning out massive amounts of ROS.
The environment literally weaponizes the immune system. I mean, we usually think of our immune cells as these independent actors fighting against external threats. But here, the physical landscape itself is dictating their behavior. It's turning them into factories of destruction.
Okay, let me synthesize this really quickly before we move to the third step, because this is getting crazy. Recap bullet 1. The chemokines pull in the CD163 positive macrophages. Bullet 2. The stiff ECM triggers FAK and NOx in the macrophages, turning them into ROS-producing factories.
Spot on.
So my question for everyone listening is, what stands out to you about the immune system being weaponized by the very physical landscape it's trying to navigate? It feels like a betrayal by your own body.
It really is. It's a physiological trap.
Yeah. But here is the piece of the puzzle I keep getting stuck on. ROS, reactive oxygen species, it's incredibly volatile, right?
Extremely. It reacts with almost anything instantly.
So how does it actually travel from the corrupted macrophage all the way over to the innocent epithelial cell to mutate its DNA? Like, it shouldn't be able to survive the trip across the empty space.
And you've hit on the exact mechanism that makes this discovery so profound, because you're right. The ROS doesn't travel. It is way too unstable.
Okay, so what does happen?
Instead, the ROS acts locally. It immediately attacks the macrophage's own outer shell. It oxidizes the lipids, the fats, that make up the macrophage's own cell membrane.
Oh. It's basically rusting its own skin.
Exactly. This process is called lipid peroxidation. The ROS attacks the membrane, creating these oxidized phospholipids, or OXPL.
FPLs, okay.
But these oxidized lipids are unstable too. So as they break down, they release secondary chemicals. And these secondary chemicals are the true long-range weapons. They are highly stable, diffusible, reactive aldehydes.
Diffusible meaning they can drift away.
Yes.
Okay, the research mentions 3 specific aldehydes, and I was looking at my notes earlier, and I actually came up with a little mnemonic trick for this.
Oh, let's hear it.
The specific aldehydes are H&E, MDA, and acrolein. So I just think of them as HMA, highly mutagenic agents.
HMA. I like that. That works perfectly because it describes exactly what they are. Unlike the initial burst of ROS, these HMA aldehydes are stable enough to actually leave the macrophage. They cross the extracellular space between the cells, and they easily seep right through the membrane of the original epithelial cell.
Okay, so they act like a toxic invisible smoke. The corrupted macrophage is burning itself up, and the smoke drifts over and suffocates the surrounding epithelial cells.
Exactly.
And once that toxic aldehyde smoke gets inside the epithelial cell, what is it actually doing to the DNA? How does it cause the mutation?
It physically disfigures it. The aldehydes chemically react with the DNA strands, forming what are called DNA adducts.
DNA adducts, what does that look like?
Well, you can visualize an adduct as this bulky, toxic chemical lump that has just been superglued directly onto the elegant double helix of the DNA.
Oh, man. So if the DNA is the cell's instruction manual, this is like spilling thick black ink over entire pages. The cell's machinery just can't read it anymore.
Yeah, but the cell certainly tries to fix it. It recognizes the damage immediately and deploys a system called NER, nucleotide excision repair.
NER?
Yeah. This is a complex biological machinery designed to basically locate these chemical lumps, cut out the damaged section of the DNA strand, and patch it with fresh nucleotides.
That sounds like a good defense.
Yeah.
But the toxic smoke is relentless, isn't it?
It is. The macrophage is constantly pumping out these aldehydes. The sheer volume of DNA adducts forming in the epithelial cell just completely overwhelms the NER repair machinery.
Oh, no.
Yeah, the repair proteins are trying to cut out the damage, but there are too many lumps. Under this immense chemical stress, the repair process fails catastrophically and the DNA strands physically snap. You get DSBs, double-strand breaks.
The genome is literally shattered.
Completely shattered.
And a shattered genome that the cell can't accurately put back together? I mean, that is the exact starting point for a cancer mutation.
Exactly. But wait, I want to add one fascinating detail about the macrophage rusting its own skin, the GPX4 detail.
Oh, right, yeah. But because there is a bizarre contradiction here, isn't there?
Yes. Usually in cell biology, if a cell's membrane undergoes extreme lipid peroxidation, like if it rusts too much, the cell just self-destructs.
Right. It triggers a specific programmed cell death called ferroptosis. I know driving cancer cells into ferroptosis by overwhelming their antioxidant defenses is actually a major area of current cancer research. Cells have built-in limits for how much membrane oxidation they can survive.
Right. So logically, the macrophage, which is generating massive amounts of ROS and rusting its own membrane to make this toxic smoke, it should just die.
The problem should solve itself. The arsonist should burn up.
It should. But Hayward and the team discovered something deeply sinister about these specific tumor-associated macrophages. They found that if they genetically knock out an enzyme called GPX4...
And GPX4 normally protects against this kind of rusting, right?
Yes. In any other situation, lacking GPX4 would cause the cell to rapidly die via ferroptosis. But these macrophages didn't die.
Wait, really? They somehow tolerate the extreme rusting without self-destructing.
They do more than tolerate it. Because they don't die, they just continue to exist as hyperactive factories, channeling even more of those toxic aldehydes outward. They have somehow adapted to survive their own destructive behavior.
That is just a devastating biological loophole.
It really is. It ensures a continuous flow of genotoxic smoke into the surrounding tissue.
Man. Okay, let's do a quick 5-minute recap of this chemical weapon phase because it's heavy. Bullet 1. ROS causes lipid peroxidation, rusting the macrophage membrane and creating OSPLs. Bullet 2. OXPLs release our HMA aldehydes HNE, MDA, and acrolein. Bullet 3. These aldehydes drift over, cause DNA adducts, overwhelm the NER system, and cause double-strand breaks.
That's the exact mechanism.
So, a question for the listener. If you were a scientist trying to stop this drifting toxic smoke, at what step of the relay would you try to intervene? Because that actually leads us perfectly into the final segment of our deep dive. The proof.
Right, the intervention.
Yeah, because we've tracked this from the initial squeeze to the panic signal to the corrupted immune cells to the rusting membrane to the toxic smoke, all the way to the shattered DNA. It's this incredibly long and complex relay. How do you even begin to prove that this entire sequence is actually happening in a living organism?
Well, you have to break the chain. If this relay is truly responsible for the DNA damage, then theoretically, if you block it at any single point, the damage should stop.
Like a process of elimination.
Exactly. Right. And that is exactly what the researchers did in Figure 1 of their paper. They didn't just observe this, they mechanically intercepted the process at 4 distinct stages to prove causality.
Okay, let's walk through these 4 interventions. If the theory is that the physical stiffness starts everything, then the first logical step would be to see what happens if you just, soften the tissue.
Right. So for their first intervention, they used compounds called BAPN, or a LOX inhibitor. These essentially prevent the cross-linking of the collagen. They relax the extracellular matrix, taking away the stiffness.
And the result?
The epithelial cells stopped tensing up, the panic signal ceased, and the DNA damage plummeted. The tissue was still there, but because it was soft, it was no longer mutagenic.
Okay, intervention 1 works. But what if you leave the tissue stiff, but you intercept the next step? The epithelial cells are still screaming for help. What if you just remove the corrupted first responders?
That was intervention 2. They used anti-CSF1, which is a targeted antibody, to selectively deplete the tumor-associated macrophages from the tissue.
Okay, so take out the TAMs.
Yeah. The tissue was still stiff, and the epithelial cells were still panicking, but because there were no macrophages arriving to become corrupted, there were no ROS factories. And again, the DNA damage vanished.
This is such elegant science. They are just isolating every single variable.
It's beautiful work.
So intervention 3 would be targeting the chemical launchpad itself, right? The rusting membrane on the macrophage.
Right. For this, they used something called EO6-SCFE.
EO6-SCFE.
What is that? It's a very specific antibody fragment. Instead of removing the macrophage, this antibody was designed to bind directly to the oxidized lipids, the OXPLs, on the macrophage's surface and neutralize them.
It essentially capped the rust.
Exactly. By neutralizing those oxidized lipids, they stopped the production of the downstream aldehydes. The macrophages were there generating ROS, but the toxic smoke could never form. The DNA damage dropped, and the overall tumor burden was significantly reduced.
Which leads the final intervention. Number 4, leave the tissue stiff. Leave the macrophages in place. Let them rust their membranes and produce the toxic smoke. Can you intercept the smoke before it hits the DNA?
Yes. They used a chemical scavenger called L-carnosine. Think of it as a molecular sponge that roams through the extracellular space.
L-carnosine, a sponge.
Right. Its sole job is to bind to and mop up those free-floating reactive aldehydes. The toxic smoke was being produced, but the sponge soaked it up before it could seep into the epithelial cells. And just like the other 3 interventions, intercepting the smoke completely protected the DNA from shattering.
That is just incredible. By breaking the chain at the beginning, the middle, and the end, they proved every single step of the relay.
They absolutely nailed it. And, the damage we are talking about isn't just minor wear and tear. When the researchers used p53 null mammary tumors and did whole exome sequencing, they found a very specific aggressive genomic fingerprint.
A genomic fingerprint. What did the sequencing show?
It revealed widespread chaos. They found extensive frameshift mutations, which completely scrambled the proteins a cell is trying to build. They found a high concentration of a mutational signature known as SBS17B.
SBS17B.
Which is a recognized hallmark of severe oxidative damage. And they saw an abnormal amount of T to G transversions where the DNA letters are literally swapped for the wrong ones.
Why does that specific type of genomic scrambling matter so much in the context of cancer? Like, why is it worse than other mutations?
Because it drives what we call mutational diversification. The tumor doesn't just acquire one single genetic flaw that a targeted drug might be able to exploit. It acquires a massive, diverse array of mutations.
So it's basically throwing everything at the wall to see what sticks.
Exactly. This high level of genetic heterogeneity makes the resulting tumors incredibly adaptable, highly aggressive, and notoriously difficult to treat. The tumor is constantly evolving because its baseline mutation rate is being artificially driven up by its own environment.
Which brings us to the most crucial part of this entire deep dive. How does this intricate cellular relay actually map onto human clinical reality? What does this mean for a real patient?
To understand the clinical impact, we have to look at the specific tissue models the researchers used. They utilized the Col1A1 model, which is characterized by exceptionally dense collagen-rich tissue.
Okay, the Col1A1 model.
Right. And they made a chilling observation. They could detect elevated levels of toxic aldehydes and severe epithelial DNA damage long before any actual tumors had formed.
Wait, really? The damage was happening in tissue that looked completely normal under a microscope?
Precisely. Histologically, normal cells were quietly accumulating catastrophic DNA damage purely because they existed in a physically dense neighborhood. And this directly explained one of the most significant yet poorly understood epidemiological risk factors for breast cancer: mammographic density.
Oh, wow. We have known for a long time that women who have dense breast tissue on a mammogram face a higher risk of developing cancer. But it always seemed like, a correlation without a clear mechanism. Was it just that there were more cells present, meaning more chances for a random error?
That was a super common theory. We also knew dense tissue was stiffer and harbored more macrophages. But Hayward and his team have finally provided the exact molecular mechanism. Mammographically dense breast tissue isn't just a passive collection of extra cells. It is a mechanically organized mutagenic field.
A mechanically organized mutagenic field. That gives me chills.
The physical stiffness of the dense tissue is actively recruiting macrophages, corrupting them, and forcing them to gas the surrounding normal cells with toxic aldehydes.
The tissue is essentially pre-primed for cancer and doesn't just sit in a static state, does it? The paper mentioned it actively worsens over time due to a vicious feed-forward loop.
Yeah, it's a devastating cycle. The initial stiffness recruits the macrophages and turns them into genotoxic factories. But those corrupted macrophages don't just damage DNA, they also secrete factors that promote even more collagen cross-linking in the extracellular matrix.
Oh, so the macrophages make the scaffolding even stiffer.
Exactly, and that increased stiffness recruits more macrophages, making them even more aggressive, which in turn drives more fibrosis. The inflammation and the physical stiffness constantly amplify one another. It creates the perfect self-sustaining storm for mutation and malignant progression.
Okay, time for our final recap. Bullet 1. Science can block this at 4 distinct stages, from softening the matrix with the LOX inhibitor to scavenging aldehydes with L-carnosine. Bullet 2. The genomic mark includes heavy frame shifts and SGS17B signatures. Bullet 3. Mammographically dense breast tissue acts as a mechanically organized mutagenic field. So, given that high tissue density can literally organize a mutagenic field, how might this change the way you, the listener, view preventative screenings like mammograms? It's a huge perspective shift.
Let me just step back and look at the whole scope of what we've covered today.
It's a lot to take in.
It really is. We started with the sheer mechanical tension of a rigid extracellular matrix. We explored how that physical squeeze forces trapped epithelial cells to hit the STAT3 panic button, flooding the area with chemical SOS signals. We tracked how those signals lure in immune macrophages, only for those TAMs to be corrupted by the stiff environment through the meganordox principle.
Yep.
We watched them rust their own membranes via lipid peroxidation, creating a toxic diffusible aldehyde smoke. Our HMAs that drifts into innocent cells, overwhelming their NER repair systems and shattering their DNA into double-strand breaks. And finally, we saw how this exact relay explains the clinical danger of mammographically dense breast tissue. It completely reframes our understanding of carcinogenesis. It proves that force, operating through a complex multicellular interaction, can leave a permanent, irreversible, and highly aggressive record in our genome.
But knowing the exact steps of this relay also means we finally have a roadmap for how to intervene.
Yeah.
We aren't just watching the storm happen. We now know 4 different ways to stop it. Which brings us to a final thought for you to carry with you today.
Yeah.
This study focused heavily on breast tissue and mammographic density. But if the physical stiffness of an organ can turn the body's own immune system into an endogenous mutagen, what does this mean for every other fibrotic disease in the human body?
The implications are profound. Could this exact same mechanical chemical circuit be quietly operating right now in the fibrotic tissues of a liver scarred by cirrhosis?
Right.
Or in a pancreas affected by chronic pancreatitis? Or in lungs suffering from pulmonary fibrosis? It suggests that the path to cancer in these organs might not require a traditional chemical carcinogen at all.
It changes everything. Maybe the future of cancer prevention isn't just about hunting down rogue, mutated cells or simply avoiding external toxins like smoke and radiation. Maybe the real future of preventative medicine lies in finding ways to physically soften the microscopic soil inside us. calming the tissue landscape long before the fire of mutation ever has a chance to start.
It entirely redefines what it means to create and maintain a healthy physical environment for your cells.
It's incredible. So the next time you think about what causes DNA damage, don't just limit your imagination to chemistry and physics. Remember the profound power of a physical squeeze. Keep questioning your assumptions about how your body truly operates beneath the surface, and we'll catch you on the next deep dive.